Hello, respected members of the Grant Committee. Thank you for the opportunity to present my research proposal. It's an honor to share my work with you. Today, I will be discussing my research on Zingibro-phenol, commonly known as ginger, and its potential role as a safe and effective therapy for rheumatoid arthritis. The focus of this project is to explore the anti-inflammatory effects of ginger and evaluate whether it can serve as an alternative option to conventional treatments that often carry significant side effects. This will be a randomized controlled trial study, and the population will be adult between the ages of 17 and 70 years who have active rheumatoid arthritis and are currently living in the United States. Arthritis is a chronic autoimmune disease that leads to joint pain, systematic inflammation, and long-term disability. Globally, more than 18 million people are affected, with about 1.5 million cases in the United States alone. Beyond the personal toll, rheumatoid arthritis is one of the leading causes of work disability and contributes significantly to healthcare costs. Current treatments are nonsteroidal anti-inflammatory drugs and corticosteroids that are effective in reducing symptoms. However, their long-term use is often associated with serious risks such as gastrointestinal bleeding, immunosuppression, and cardiovascular complications. Because of these challenges, there is growing interest in natural and plant-based therapies, which may offer effective symptoms relief with fewer side effects. Ginger has long been used for inflammatory conditions. Its key compounds such as 6-gingerol and shogel have demonstrated anti-inflammatory effects. Early evidence from animal and small human studies is promising, but larger clinical trials are needed to confirm its role in rheumatoid arthritis. Most preclinical studies on ginger have been in rodents or cell models, which don't fully translate to humans. Human studies so far are limited and have focused mainly on gene expression without assessing protein biomarkers. What's missing is a comprehensive randomized controlled trial that evaluates serum cytokines, uses a standardized dosage, and measures clinical outcomes like disease activity score 28 joints together. This is the critical gap my study is designed to address. Here is the visual evidence of one of the studies I reviewed, showing that ginger extract significantly reduced inflammatory cytokines like tumor necrosis factor alpha and interleukin 1 beta and 6 in a rat model of arthritis. It highlights the preclinical evidence that supports ginger's anti-inflammatory potential. So building on these evidence, my study will test these effects in humans. I am conducting a randomized double-blinded controlled trial to test the effects of ginger extract in adults with the active rheumatoid arthritis in the US. Participants will receive either ginger supplements or placebo for 12 weeks. I will measure serum cytokines and disease activity scores, and by analyzing both gene expression and protein levels, I aim to determine whether ginger can safely reduce inflammation and improve rheumatoid arthritis. I am uniquely positioned to address this important research gap by combining clinical insights into the burden of rheumatoid arthritis with a deep understanding of molecular immunology, including gene and protein level markers. And my commitment to patient-centered research ensures that this study focuses on US adult age 17 to 70 years and translating preclinical evidence into meaningful human trials. This project embodies a true bench-to-bedside approach, aiming to generate scientifically rigorous and clinically relevant evidence on the potentials of ginger supplementation to improve rheumatoid arthritis outcomes. I hypothesize that ginger supplementation will reduce key inflammatory cytokines, including tumor necrosis factor alpha and interleukin 1 beta and 6 and CRP, and improve disease activity as measured by 28 body joints. If successful, these findings could inform clinical guidelines and offering a safe and natural therapy for rheumatoid arthritis and empower patients to make evidence-based choices about managing their disease. The results of this study will be communicated to scientific community through peer-reviewed journals and conferences and guide clinicians in complementary care and reach patients via the Arthritis Foundation and social media for maximum real-world impact. This study could show that ginger safely reduces inflammation in rheumatoid arthritis, guiding clinical practice, supporting patient-centered care, and empowering patients to make informed choices that improve their quality of life. Additionally, cost-effective rheumatoid arthritis medicine will be available in the market with less or no side effects. Thank you for reviewing my proposal and considering this research. I really appreciate the opportunity to contribute to improving care for adults with rheumatoid arthritis.